Did you know that human beings as a species first showed up in Africa? But people today live all over the globe. So how did they get there? And how do scientists figure this out? Well, turns out there are lots of ways to determine how a small group of human beings in Africa spread across the world, many of which are genetic markers found in your DNA. Genetic markers give us information about the allelic variation in different loci, and there are a number of techniques and different genetic markers we can use to study variation in the human genome. For example, we have used markers known as allozymes to study protein variants. Here, enzymes of the same nature were analyzed by running them on a gel in order to distinguish polymorphisms caused by amino acid substitutions, namely size and charge differences between the proteins. To look at sequence differences, we have used markers such as RFLPs, restriction fragment length polymorphisms, and SNPs, which are single nucleotide polymorphisms. RFLP analysis, for example, uses restriction endonucleases, which are enzymes that cut DNA at specific consensus sequences throughout the genome to produce different sized DNA fragments. We can then analyze the differences in the fragment lengths produced by the cuts to observe polymorphisms between different peoples. While RFLPs were the basis for early genetic fingerprinting, advances in sequencing technology has made other methods more favorable, such as using mitochondrial DNA and SNPs. There also exists polymorphisms in certain regions of the human genome containing tandem DNA repeats, or repeats of DNA next to each other. These markers are known as microsatellites and minisatellites. Different individuals and populations have different numbers of these tandem repeats in their genomes. Microsatellites are particularly popular molecular markers due to the small size of their repeats, which is about 100 base pairs, and allows the satellite regions to be amplified by PCR for analysis. The variations in these different types of markers can help us determine migration routes of early modern humans. From genomic data, we have inferred that anatomically modern humans emerged from Ethiopia 150 to 190,000 years ago, and a single out-of-Africa dispersal event occurred 55 to 65,000 years ago. This is a migration event from which all modern non-African people branch. The out-of-Africa event led to the spread of peoples into Asia. The first wave occurred 45 to 55,000 years ago to Sahul, which is present-day Australia and New Guinea. The other wave, 36 to 45,000 years ago, included the ancestors of East Asians. Around 11,000 years ago, animal husbandry emerged, and the disbursement followed from central Anatolia into Europe. Around 15 to 23,000 years ago, ancestors of Native Americans in Siberia migrated across the Bering Land Bridge to people the Americas. Next, we will discuss 4 different types of markers used to explore human migration routes: ALU, mitochondrial DNA, SNPs, and viruses. ALU is a transposable element in our genomes, and it has the ability to tell us where we come from. That means it copies and pastes itself, jumping around the human genome and inserting itself into new places. What makes ALU important for the study of human evolution is its ability to identify lineages from a common ancestor. There are over 1 million ALU transposons in an individual, representing about 11% of a genome. The probability that ALU will insert independently into the same place more than once is practically zero. That means that 2 people who have the same ALU sequence in the same place on the genome must be related by a common ancestor. A study from 2005 analyzed allele frequencies for 12 ALU insertions in many different populations of Native Americans and Asians. They found a trend in a reduction of genetic variability from Southeast Asians to South Native Americans. The reduced genetic variation in the Americas can be attributed to the founder event, which is the loss of genetic diversity when a new population is established with a smaller number of people. This founder event, which resulted in the peopling of America, occurred 15,000 years ago during the crossing of the Bering Land Bridge. In addition, the people of the Americas commonly split into tribes, which is hypothesized to magnify the effect of the founder event with further reduction in genetic diversity. Now that we know how transposable elements can tell us about migrations, we're going to show how mitochondrial DNA markers can be used to support the Beringia standstill hypothesis. The last global maxima occurred 28,000 years ago and was a period of intense cold that led to the expansion of massive ice sheets and a large decrease in plant and animal productivity. During this time, humans from Siberia migrated to Beringia, a land bridge that used to connect what is modern-day Siberia and Canada. During local climate changes, the population became trapped in Beringia, cut off from Siberian glaciers. When the last glacial maximum ended and the glaciers receded 15,000 years ago, the population began to people the Americas. This hypothesis was formulated using mitochondrial DNA haplotypes. Mitochondrial DNA, or mtDNA, is a short, circular molecule that encodes proteins that are involved in human respiration, and it is only inherited from the mother. mtDNA mutations are considered to be clock-like, so that the amount of differences in mtDNA between populations roughly reflects the time at which the populations diverged from one another. A haplotype is a group of genes in an organism that were inherited together from a single parent, and these groups of genes can accumulate mutations that make them different from one another. Different mitochondrial haplotypes are labeled by a different letter of the alphabet and are used to show where differences emerged and populations diverged on a tree of life. Researchers that proposed the bringing standstill hypothesis compared mtDNA from Native American individuals and Asian individuals. They found that subclades C1b, C1c, and C1d incorporate nearly all of Native American haplogroup C mtDNAs, but they are absent in Asia. These subclades arose around 15,000 years ago at the time when the ancestors of Native Americans began to people the Americas. This suggests that the population that would people the Americas paused in Beringia long enough for mutations to accumulate that are completely separate from their Asian ancestors. Another method of tracking ancestry and migration is through the use of genome-wide SNP genotyping. SNPs, or single nucleotide polymorphisms, are variations in a single base pair at a specific place in the genome. These can be inherited and passed on from generation to generation and become prevalent in populations. When populations are separated for long enough time, they would begin to show different SNP makeups. Looking at these makeups today can show us where different populations came from and if some populations were originally formed by migrating away from a larger one. For example, genome-wide SNP genotyping on modern-day Native Americans has shown 3 different ancestral groups formed from 3 different gene flow events from Asia to the Americas. These gene flow events could have themselves been migrations. Findings such as these stand in contrast to the standstill model and favor 3 migratory waves across the Bering Land Bridge. So now we understand how we can look inside our own DNA to see where our ancestors have been. But did you know that infectious agents can also tell us our past? Viruses and bacteria can be looked at to see how humans have migrated. Some viruses and bacteria co-evolve with their host, which means that as we change over time, the viruses and bacteria change as well. This is what allows us to look at these agents instead of our own genomes to see migration. A common way of doing this is by sequencing either snippets of their genome or the whole genome. It is easier to do this than sequencing a whole human genome, since viruses and bacteria are much smaller. An example of this is the HSV-1 virus that causes cold sores. HSV-1 has always been around, but it is not fatal, which means that it is a good virus to look at when trying to understand human migration. After sequencing the genome, you can compare the difference in sequences to build a family tree for the virus by looking at HSV-1 genomes and comparing them to known HSV-1 sequences from all over the world. From there, you can determine human migration. This is used to determine migration events from a very long time ago. HSV-1 family tree gives evidence supporting the theory of human migration starting in Africa. Another way to use infectious agents to look at migration is by looking at the antibodies present in humans. An antibody is a part of our immune system that finds and attaches to infectious agents that are in your body. There are many different types of antibodies, and each antibody only recognizes a specific protein found on the agent. This specificity makes it possible to look at the differences in antibodies in different populations and track the changes to see how people have moved. This approach was used to see migration into Brazil, from North America after crossing the Bering Strait, and also from Africa and Japan. So hopefully now you have learned a little bit more about genetic markers and their roles in predicting human migration patterns. Thank you for watching!